The transduction pathway for the visual system involves the transfer of stimuli through a complicated network of neurons. Stimuli travel through the photoreceptors, horizontal cells, bipolar cells, and retinal ganglion cells. The retinal ganglion cell axons then travel through the optic chiasm and synapse onto the lateral geniculate nucleus axons. LGN axons then project up to layer 4 in the cortex. Transduction begins when a stimulus hits the photoreceptors. Although it would be intuitive to think that light is the stimulus that leads to depolarization, the photoreceptors actually hyperpolarize in response to light. They depolarize and release glutamate when there is an absence of light. The amount of glutamate released is dependent on the amount of depolarization in the photoreceptor. Multiple photoreceptors then feed into a single bipolar cell. The cluster contains both center photoreceptors, which feed directly into the bipolar cells, and surround, which link up to the intermediary horizontal cells. Horizontal cells, which release an inhibitory neurotransmitter when depolarized, synapse on the presynaptic terminal of the photoreceptor. There are two types of bipolar cells: off-center bipolar cells and on-center bipolar cells. The two types of cells have two different types of receptors. Off-center bipolar cells, which have AMPA receptors, depolarize in response to a glutamate signal, while on-center bipolar cells, which have a metabotropic glutamate receptor, hyperpolarize in response to a glutamate signal. The input from both the photoreceptors and the horizontal cells will give the input that will lead to the level of depolarization or hyperpolarization seen in the two types of bipolar cells, which then determines the amount of glutamate that will be released from the bipolar cells to the retinal ganglion cells. The retinal ganglion cells then depolarize in response to glutamate. In an off-center bipolar cell, when there is no light in the center but light on the surround photoreceptors hyperpolarize in response to light. The center photoreceptor depolarizes in response to dark or absence of light. This in turn results in the horizontal cells hyperpolarizing. This means the horizontal cells cannot release inhibitory neurotransmitters. The off-center bipolar cell then depolarizes in response to glutamate. When the off-center bipolar cell depolarizes, the retinal ganglion cell depolarizes as well, sending a signal onward. Light on the center and no light on the surround photoreceptors depolarize in response to absence of light. The center photoreceptor hyperpolarizes in response to light. This results in the horizontal cell depolarizing and releasing inhibitory neurotransmitters, further limiting the amount of glutamate getting to the bipolar cell. The off-center bipolar cell then becomes hyperpolarized, and therefore the retinal ganglion cell does not depolarize. In an on-center bipolar cell, when there is light on the center and no light on the surround photoreceptors depolarize, and the center photoreceptor hyperpolarizes. This leads to the horizontal cell depolarizing, which causes it to release an inhibitory neurotransmitter that prevents the center photoreceptor from releasing its glutamate. As a result, the on-center bipolar cell becomes depolarized, which in turn causes the retinal ganglion cell to depolarize. On the other hand, when there is light on the surround and no light on the surround photoreceptors hyperpolarize and the center photoreceptors depolarize. This in turn causes the horizontal cell to depolarize and be unable to release the inhibitory neurotransmitter on the photoreceptor. So, the center photoreceptor releases glutamate and causes the on-center bipolar cell to become hyperpolarized, which in turn leads to the retinal ganglion cell not depolarizing.